The abnormalities or the change in the chromosomal number will lead to anomaly or abnormality, or otherwise we call it as syndromes. Hemodialysis, the very word itself will tell you, blood. UGG codon codes for tryptophan. The keystone species is the species that has a disproportionately large effect on its environment relative to its abundance. Hello everyone, welcome back to 2010 Karnataka CET Biology Paper Analysis, that is KCET 2010 Biology Paper Analysis. So far we have completed up to 2011 and hope you have followed all the analysis part in this biology paper. So in today's session, I'll be dealing with Karnataka CET 2010 biology paper analysis. Moving on to question number 1. Due to non-disjunction of chromosomes during spermatogenesis, sperms carry both X chromosomes, 22A plus XY, and some sperms do not carry any sex chromosome, that is 22A plus O. If these sperms fertilize normal X, that is 22A plus X, what type of genetic disorders appear among the offsprings? Option A, Down syndrome and Klinefelter syndrome. Option B, Turner syndrome and Klinefelter syndrome. Option C, Down syndrome and Cray-Duchat syndrome, and Down syndrome and Turner syndrome. As we know, the abnormalities or the change in the chromosomal number will lead to anomaly or abnormality, or otherwise we call it as syndromes. It may be due to monosomy 2n-1, or it may be due to trisomy, that is 2n plus 1. And that monosomy or trisomy may be to an autosome or to an allosome. So we call it as autosomal disorders and allosomal disorders. So Down syndrome is an autosomal disorder and Klinefelter syndrome is an allosomal disorder. Turner syndrome is an allosomal disorder, and as well as Klinefelter syndrome is an allosomal disorder. Down syndrome and Cray-Duchat syndrome are autosomal disorders. So Down syndrome is autosomal disorder and Turner's syndrome. Turner syndrome. Here, Turner syndrome occurs due to monosomy 2n-1, and that -1 is to the allosome. Usually we find it in the females. So the chromosomal complement will be 44A plus X0, that is 45. Here the nucleus of the cell will have 45 chromosomes. So it is having unbalanced set of chromosomes, that is 45 chromosomes instead of 46. That is 22A plus X0, that is 22A plus 0, right? So Klinefelter syndrome is due to trisomy, and that +1 is added to the allosome. It is formed by the union of an abnormal XX egg and normal sperm, or normal X egg and abnormal XY sperm. The individuals will have what? 47 chromosomes. So what will be the chromosomal complement? It will be 22A plus XY. It will be 22XY actually. So instead of 46 chromosome, it will be 47 chromosome. So it is, so what type of genetic disorders appear among the offsprings? It is Turner syndrome and Klinefelter's syndrome. So option A, Down syndrome and Klinefelter syndrome is wrong. Turner syndrome and Klinefelter syndrome is correct. Down syndrome and Cray-Duchat syndrome is wrong. Both are allosomal disorders. So here both are allosomal disorders. So here one is autosome and one is allosome. So this is also wrong. So option B is correct. Option B is correct. Moving on to question number 2. When a freshwater protozoan is placed in marine water. Option A, the number of contractile vacuole increase. Option B, the contractile vacuoles become bigger in size. Option C, the contractile vacuoles remain unchanged. And option D, the contractile vacuoles disappear. When a freshwater protozoan is placed in marine water, the contractile vacuoles disappear. The very important function of contractile vacuole is to what? To remove the excess of water outside by the process of osmoregulation. And in marine water, it is necessary to retain the water inside the body. So usually in marine water, the organisms have to retain the water inside the body instead of expelling out. If contractile vacuoles will not disappear, then the cell will get dehydrated. So the contractile vacuole disappear. So when a freshwater protozoan is placed in marine water, the contractile vacuole disappear. So option A, the number of contractile vacuoles increase is wrong. Option B, the contractile vacuoles become bigger in size is wrong. Option C, the contractile vacuoles remain unchanged is wrong. Option D, the contractile vacuoles disappear is correct. Option D is correct. Question number 3. Which one of the following pairs is an example for lateral meristem? Option A, interfascicular cambium and phellem. Option B, procambium and phelloderm. Option C, phellogen and fascicular cambium. And option D, phellogen and phelloderm. Lateral meristem, as you know that lateral meristem, these meristems are known as vascular and cork cambium, and they are active in the areas of the plant where primary growth has ceased. So they are active in the area where primary growth has ceased and are therefore referred to as what? Secondary meristems. So the lateral meristem encircle the body of the whole plant, and phellogen is defined as the meristematic layer responsible for the development of periderm. So it is phellogen and fascicular cambium. So option A, interfascicular cambium and phellem is wrong. Option B, procambium and phelloderm is wrong. Phellogen and fascicular cambium is correct. And phellogen and fascicular cambium. D, phellogen and phelloderm is wrong. So option C is correct. Option C is correct. Moving on to question number 4. In peritoneal dialysis, option A, the blood is not removed from the body and a natural filter is used. Option B, the blood is removed from the body and a natural filter is employed. Option C, the blood is removed from the body and an artificial filter is employed. Option D, the blood is not removed from the body and an artificial filter is used. In peritoneal dialysis, as we know, what is dialysis? The process of filtering the blood when the kidney is not functioning properly. So the filtration of the urine will not take place, or a filtration of the blood will not take place. So the process of filtering the blood by the artificial kidney is known as dialysis. So artificial kidney here is nothing but a dialysate, a filter. Right. So in peritoneal, there are two types of dialysis. One is called peritoneal dialysis, the other one is called hemodialysis. So hemodialysis, the very word itself will tell you, blood. So here, peritoneum of the abdomen, an incision is made and a catheter is introduced. So in peritoneal dialysis, the peritoneal dialysis involves what? A surgery to implant a peritoneal dialysis, that is dialysis catheter, into the abdomen. So the catheter will help to filter the blood through the peritoneum, which is a membrane. So a filter. So here the blood is not removed from the body and a natural filter is used. So the blood is not removed from the body and natural filter is used is correct. Whereas in hemodialysis, the blood is removed from the body. So here option B, the blood is removed from the body is wrong. The blood is removed from the body and an artificial filter is employed is wrong. Option D, the blood is not removed from the body and an artificial filter is used is wrong. So option A is correct. Moving on to question number 5. The following is a scheme showing the electron transport system, ETS system. Identify the electron carrier molecules indicated as A and B and choose the correct option. So here you can see it is not so very clear, but here A and this is B. Here you can see cytochrome B1, C1, cytochrome oxidase complex, right? So here in ETS system, we find that NADH passes electron to flavoprotein, to a series of iron sulfur protein, and finally to ubiquinone, that is coenzyme Q. So here coenzyme Q, that is A. So A is coenzyme Q and is a lipid soluble benzoquinone. It is a coenzyme Q is a lipid soluble benzoquinone that serves as electron carrier and transfer the electrons to cytochrome C1, BC1 of complex 3. Cytochrome C is which is represented by B. So A is coenzyme Q and B is cytochrome C is a soluble protein that accepts an electron from complex 3 and donate them to the binuclear copper center of complex 4. So A is coenzyme Q and B is cytochrome C. So option A cytochrome C, B coenzyme wrong. A coenzyme Q correct, B cytochrome C is correct. So option B is correct. This is FMN, FES protein is wrong. FES protein, FMN is wrong. So option B is correct. Moving on to question number 6. The sugar present in milk is option A sucrose, option B fructose, option C lactose, and option D glucose. As we know, the sugar present in milk is lactose sugar. Lactose is what? It is a disaccharide sugar derived from what? From monosaccharides called galactose and glucose. So lactose makes up about around 2 to 8% of the total weight of milk, and usually we find that the infants of mammals feed on the milk which is rich in lactose, which is later digested by the enzyme called lactase in the intestine. So the sugar present in milk is lactose. So sucrose it is found in, right? So option A sucrose is wrong, option B fructose is wrong, option C lactose is correct, option D glucose is wrong. So option C is correct. Moving on to question number 7. According to Stewart's starch hydrolysis theory, which one of the following is the principal reason for the opening of the stomata during daytime? Option A, conversion of sugar into starch in guard cells. Option B, influx of potassium ions into guard cells under the influence of ABA hormone, abscisic acid hormone. Option C, photosynthetic utilization of carbon dioxide in guard cells. Option D, efflux of potassium ions from guard cells under the influence of ABA hormone. Here, this Stewart starch hydrolysis theory is a classical theory of stomatal opening and closing, which was proposed by Sayre in 1923. Here, the guard cells contain starch, which is hydrolyzed to form glucose under high pH caused due to reduced carbon dioxide concentration. So, their starch guard cells contain starch, which is hydrolyzed to form glucose, and a high pH is caused. Why? Due to reduced carbon dioxide concentration. Why there is a reduction in the carbon dioxide concentration? Because it is utilized for photosynthesis. So the carbon dioxide concentration is reduced for the process is due to the process of photosynthesis. So glucose increases the osmotic concentration and hence DPD is caused, that is diffusion pressure deficit of guard cells. So what will happen? The lateral absorb water from the epidermal cells, swell up, the guard cells will swell up and open the stomatal pore. So that is what is the theory, the principle that is involved in for the opening and opening of stomata during daytime is photosynthetic utilization of carbon dioxide in guard cells. So option A, conversion of sugar into starch in guard cells is wrong. Influx of potassium ions into guard cells under the influence of ABA hormone is wrong. Photosynthetic utilization of carbon dioxide in guard cells is correct. Option D, efflux of potassium ions from the guard cells under the influence of ABA hormone is wrong. So option C is correct. Moving on to question number 8. Which one of the following process results in the formation of a clone of bacteria? Option A, conjugation. Option B, binary fission. Option C, transduction. And option D, transformation. We all know that binary fission is a type of asexual reproduction in which the parent cell divides and reproduces in such a way that the parent cell divides, the contents of the parent cell divide and form two daughter cells, right? So the nucleus divides and then the cells divide to produce two cells. The daughter cells are produced from a single parent and contain the exact copy of genes as the mother. So they are considered as what? Clones. Clones are nothing but they are exact copy of their parents. So which are genetically similar cells. So which one of the following processes results in the formation of a clone of bacteria? It is binary fission. So option A, conjugation, is a type of sexual reproduction. Binary fission is asexual reproduction. Transduction is a type of sexual reproduction. Wrong. Transformation also is wrong. So option B is correct. Moving on to question number 9. Match the types of immunity listed in column 1 with the example listed in column 2. Choose the answer that gives the correct combination of alphabets of the two columns. So column 1 contains the immunity and the column 2 contains the examples. So natural active, artificial passive, PASSI, artificial active and natural passive. So natural active is fighting infections naturally. Artificial passive, injection of antiserum to travelers. Then artificial active, induced by vaccine. Then natural passive, from mother to fetus through placenta. So A, S, B, R, C, T, D, Q. So A, yes, first one it should be AS. Here it is AT. So obviously this is wrong. Here it is AS. Okay. Here also it is AS. Here it is APU. So it is wrong. Go for the next. BR. Here it is BT. So it is wrong. Here it is BR. Correct. CT. Correct. Here also CT. DQ. Here also DQ. So option C is correct. Natural active, fighting infections naturally. Artificial passive, injection of antiserum to travelers. Artificial active, induced by vaccination. Then natural passive, from mother to fetus through placenta. So option C is correct. Moving on to question number 10. How do you differentiate a butterfly from a moth? Option A, moth has one pair of wings, but butterfly has two pair of wings. Option B, moth has feathery antennae, but butterfly has club-shaped antennae. Option C, moth has simple eyes, but butterfly has compound eyes. Option D, moth is diurnal, but butterfly is nocturnal. So usually, a moth antennae is feathery, whereas the butterfly's antennae are club-shaped. Moths are nocturnal, flying at night, whereas even butterflies are primarily, they are also nocturnal and they are also nocturnal as well as diurnal. So moth has one pair of wings, but butterfly has two pair of wings is wrong. Moth has feathery antennae, but butterfly has club-shaped antennae. Correct. Moth has simple eyes, but a butterfly has no. Both have compound eyes. Moth is diurnal and butterfly is nocturnal is wrong. So option B is correct. Option B is correct. Moving on to question number 11. Which one of the following statement is not correct during protein synthesis? Tyrosine is coded by UAU and UAC codons. Cysteine is coded by UGU and UGC codons. Then UGG codon codes for tryptophan, UAA codon codes for lysine, which is not correct. During protein synthesis, we find UUA codon codes for leucine, and UGG codon codes for tryptophan. So here it is correct. UGG codes for tryptophan is correct. Then cysteine is coded by codon EU, GU and UGC. This is also correct here also. Then tyrosine is coded by two codons UAU and UAC. Here UAU and UAC also is correct. So which one of the following statement is not correct? UAA codes for lysine is not correct. Lysine, that is AAG, AAA codes for lysine. So UAA codon codes for lysine is wrong. So option D is wrong. Moving on to question number 12. In the absence of acrosome, the sperm cannot get energy. Option A. Option B, cannot penetrate egg. C, cannot swim. D, cannot get food. As we know, the acrosome is a very, very important the cap-like structure of the sperm, which helps the sperm to get through the egg's protective coat and to allow the plasma membrane of the sperm and the egg to fuse. So if there is no acrosome, there will be no entry of the sperm nucleus into the egg. So if it is the acrosome which is responsible for sperm entry. So option A, cannot get energy, is wrong. Option B, cannot penetrate egg, is correct. So without which it cannot penetrate egg. C, cannot swim, is wrong. Option D, cannot get food, is wrong. So option B is correct. Option B is correct. Moving on to question number 13. The diagram of the ultrastructure of a plant cell is given below. Identify the functions of the organelles labeled A, B, C and D and E in the diagram. So A, it is very much the director of macromolecular traffic, we can say. So it is Golgi body. Then B, mitochondria. So as you know, mitochondria is the site of oxidative phosphorylation. And C, vesicle, that is intracellular transport. Then D, site of protophosphorylation. Then E, storage of cell sap, that is vacuole. You can see the central vacuole, storage of cell sap. So option A, principal director of macromolecular traffic, is correct. B, site of, principal director of macromolecular, that is Golgi body. Then B, site of oxidative phosphorylation, mitochondria. Intracellular transport, that is vesicle. D, site of protophosphorylation, that is chloroplast. And E, storage of cell sap, is the central vacuole. So option A is correct. So option A is correct. Here you can see intracellular transport, wrong. Here we can see storage of cell sap, wrong. Here's site of photophosphorylation. The first A itself is wrong here. You can see, right? So option A is correct. Option A is correct. Moving on to question number 14. Which one of the following species of earthworm is not recommended for vermicomposting? I think all of us know that earthworms are called farmer's friend, and this very important process called vermicomposting will produce a very good fertile, we can call it as organic manure. So there are certain species of earthworms which are used for vermicomposting. So all earthworms are not used for vermicomposting, as we classify the earthworms into two types. One called humus formers. They form the humus. Humus is nothing but the fertile soil, right? Then the other group of earthworms are called humus feeders. So among the humus formers are Eurydilis eugeniae, Eschenia foetida, then Perionyx excavator. These are all called humus formers. They form the humus. But Peritima posthuma earthworm is not recommended for vermicompost because it is a humus feeder. So among the four, which one of the following species of earthworm is not recommended? So Yersinia foetida is recommended. Uridillus eugenia, it is recommended. Then Peritima postema, it is not recommended. So Perionyx excavate, it is recommended. So the one which is not recommended for vermicomposting is Peritima postema. That is Peritima postema. So option C is correct. That is the one which is not recommended for vermicomposting. Moving on to question number 15. The main aim of HGP, that is Human Genome Project. Option A, to identify and sequence all the genes present in human DNA. Option B, to introduce new genes into humans. Option C, to remove disease-causing genes from human DNA. And option D, to develop better techniques for comparing two different human DNA samples. I think all of us know human HGP, that is Human Genome Project, aims at determining the sequence of nucleotides, that is the DNA, that make the whole human DNA. So it is mainly involved in sequencing the human DNA. So genome sequencing has its application in identification of disease-causing genes and their elimination, to introduce new genes that code for desired proteins, products, and to exploit polymorphism to compare DNA samples from two individuals. So option A, to identify and sequence the genes present in human DNA is correct. To introduce new genes into human, also correct, but this is the main thing. To identify and sequence all the genes present in human DNA. So to remove disease-causing genes from human DNA, to develop better techniques for comparing two different human DNA sampling. So the one which is correct is to identify and sequence all the genes present in human DNA. So option A is correct. Moving on to question number 16. The species, though insignificant in number, determine the existence of many other species in a given ecosystem. Such species is known as option A, sacred species, option B, endemic species, option C, keystone species, and option D, extinct species. We know that the keystone species is the species that has a disproportionately large effect on its environment relative to its abundance. So, the main structure of an ecological community helps the types and the number of various other species in the community. So, the species, though insignificant in number, determine the existence of many other species in a given ecosystem. Such species is known as keystone species. So option A, sacred species is wrong. Option B, endemic species is wrong. Option C, keystone species is correct. And option D, extinct species is wrong. So option C is correct. Moving on to question number 17. Compare the statements A and B. Statement A. Sclerenchyma cells do not have plasmodesmata. Statement B. The cell walls of some permanent tissues are heavily lignified. Correct. I think all of us know that in simple permanent tissues, we have parenchyma, collenchyma, and sclerenchyma. Sclerenchyma is called as a dead mechanical tissue, which is heavily lignified with, as you know, it gives mechanical support because of its lignified cell walls. And plasmodesmata are nothing but called as cytoplasmic channels, which communicate with each other. So plasmodesmata are narrow channels that act as intercellular cytoplasmic bridges to facilitate communication and transport of materials, which is not found in sclerenchyma because it is a dead mechanical tissue and it is heavily lignified. So both the statements are correct. So option A, both the statement A and B are wrong is wrong. Option B, statement A is correct and B is wrong is wrong. Option C, statement A is wrong and B is correct is wrong. Both the statements A and B are correct is correct. So option D is correct. Moving on to question number 18. Which one of the following is not the reason for very high load of bilirubin? As we know, bilirubin is a bile pigment, usually which causes jaundice in children. High load of bilirubin causes jaundice. But which one of the following is not the reason for very high load of bilirubin in newborn? The liver of newborn is too young to cope up with the heavy load of bilirubin. Option B, excessive red blood corpuscles in the newborn burst, releasing the bilirubin. Option C, insoluble bilirubin in the intestine is reabsorbed by the blood. Mother's milk contain a high amount of bilirubin. You know, during the lactation period, when the mother is feeding the young one or the newborn, as you know, the milk contains colostrum, which acts as an antibody for the newborn in future course of its lifetime, and it has high content of bilirubin, but which is not going to cause any threat to the newborn. Instead, it is going to protect the newborn. So, which of the following is not the reason for very high load of bilirubin in a newborn? This will not cause high load of bilirubin in the newborn. That is not the reason. Milk is not the reason for the high load. So, the liver of the newborn is too young is wrong. Then excessive red blood corpuscles in the newborn releasing bilirubin is wrong. Insoluble bilirubin in the intestine is wrong. Mother's milk contain high amount of. So, this is not the reason for mother's milk contain high amount of bilirubin is not the reason for very high load of bilirubin in the newborn. So, option D is correct. Moving on to question number 19. Which one of the following diseases is caused by Nosema bombesis in mulberry silkworm? Option A, pebrine. Option B, muscadine. Option C, that is flagellate. And option D, gracillate. Option D, gracillate. So, we know that pebrine is a disease of silkworm which is caused by a microsporidium called Nosema bombesis. And this is going to kill all the silkworm hatched from infected eggs, which is got through the food that the silkworm eats. So, which one of the following disease is caused by Nosema bombesis? It is pebrine. So, option A, pebrine is correct. Option B, muscadine is wrong. Option C, flagellate is wrong. And option D, gracillate is wrong. So, option A, pebrine is correct. Moving on to question number 20. The following is a scheme showing the path of reflex arc. Identify the different labeling A, B, C, D, E, F in the reflex arc. Here you can see the reflex arc A, B, C, D, E, F. So, you know, reflex arc is nothing but a spontaneous, rapid and predictable motor response to stimulus. So, A to C, we can call it as shows the receiving of the stimulus, and D to F, this is shows the response to that particular stimulus. So, this is receiving end and this is responding end. This is response to external stimuli. So, here, so what is A? We can see that A is nothing but stimulus. So, A is stimulus, here also A is stimulus, here also A is stimulus, here is stimulus. Then B is receptor, A is stimulus, B is receptor. So, here also we come across receptor, here B is effector, so this is wrong, here receptor, here B is effector, wrong. So, we have to identify in that. Then C is, here we can see C is sensory nerve, C is sensory nerve. A is stimulus, B is receptor, C is sensory nerve. Here we can see C is sensory nerve, correct. Here C is motor nerve is wrong. Then D is motor nerve. D is motor nerve. So here we can see D is motor nerve. Here C is sensory nerve. Here also C is sensory. But this is wrong already. So this is correct. E is effector and F is response. Here we can see E is effector and F is response. So A stimulus, correct. B receptor, correct. C sensory nerve, correct. D motor nerve, correct. E effector nerve. F responses. So option A is correct. Hope you have understood all the 20 questions that I have analyzed in this 2010 KCET biology paper. So I'll be back with some more questions. Till then, stay tuned.